Good afternoon, good morning, good evening, whatever time it is for you. Hello, it's Man Big T, Talent Talks, the biology believed by Dr. Bruce Lipton. We're making our way. We're almost halfway. I hope what I'm reading you're retaining. I know there's a lot of scientific jargon in here. It's probably one of the biggest things that I've had to overcome when it comes to learning or reading some of the certain books that I've decided to purchase and dive into is understanding all the scientific jargon and everything that they're trying to explain when you have no background in said field. So, anyways, we're carrying on where we left off. I believe we're still chapter 4. We're almost on chapter 5. 3 pages. Yeah, and we're carrying where we left off. The researchers concluded that those 3 signaling mechanisms are clearly not necessary for chili seeds and basil plants to sense each other's presence. The authors of the study state that no mechanistic explanation of how plants may perform the observed feat is yet available. But they go on to explain that such a communication channel requires the emission of a signal that not only propagates rapidly to convey real-time information about neighboring plants, but also can be analyzed quickly. Sounds like quantum energetic communication to me. Sounds like quantum energetic communication to me. Quantum magic is also being uncovered in studies of one of life's most important biological activities, photosynthesis. I hope you remember what that is. Photosynthesis involves the capture of a photon light by chlorophyll, a multi-protein complex, and how the energy of that light is used to mobilize electrons that empower the creation of organic chemistry from inorganic CO2 and water. The electrons transferred in the photosynthesis process can choose among many different pathways as they travel through the protein complex. However, because photosynthesis is a highly efficient process, electrons ultimately choose to utilize a single pathway. But how? Quantum mechanics, in the form of superposition, enables a quantum particle, an electron in this case, to simultaneously experience all possible pathways and then choose the most effective one on which to travel. Whoa, that's wild. The photosynthesis process requires a specific and precise alignment of the molecular components in the complex in order to effectively steer this reaction to its successful conclusion. In 2006, Canadian physicists and chemists demonstrated that manipulating vibrational frequencies and exploiting the wave-like nature of matter can selectively direct the behavior of atomic and molecular systems. But chlorophyll proteins work at a relatively high ambient temperatures that cause these molecules to experience random thermal vibrations. How are electrons efficiently shuttled through the chlorophyll complex in the presence of these random vibrations? Combining molecular dynamics and quantum chemistry to study electron transfer processes, physicists at the University of California, San Diego offered a profound solution to this problem. Their study revealed that electron transfer occurs through a web of quantum tunneling pathways created through constructive or deconstructive interference patterns typical of wave-like processes of quantum mechanics I described in this chapter. The challenge with all this new research is that while it is an indisputable fact that immaterial, energetic signals can control biology, there is no conventional known mechanism to explain such phenomena. Well, at least there is no known classical Newtonian mechanism to account for this behavior. In our current world, information must be measurable before it becomes real or accepted by the scientific community, and a major problem in studying the biological influence of quantum signaling is that it is difficult to measure this energy. Frequently, this form of communication is so subtle that the amplitude of life-influencing signals is way below the resolution of scientific instrumentation. In 2014, an international research team illuminated the fact that cellular signaling by the Ross family of membrane proteins, one of the most important components of signaling networks in biology, is so subtle that it cannot be distinguished from background noise. That experimenters conventionally disregard as a technical artifact. Energy fields influencing cell membrane receptors, such as those controlling RAS proteins or RAS proteins, represent the interface wherein environmental signals control cell functions. Experimental manipulation of environmental EMF, electromagnetic field, frequencies has been shown to profoundly influence the activity of a cell's sodium, Na+, potassium, K+, ATPase, and calcium, Ca2 plus ATPase, ion protein channels. Since these membranes proteins control the cell's electrical activities, including the development and maintenance of the cell's membrane potential, environmental electromagnetic fields can shape the health and fate of biological systems. For example, it is now well established that microwave radiation associated with cell phones and other electronic devices interferes with and disrupts normal cell behaviors and can possibly lead to dysfunction and disease. Of course, as always in science, there were a number of quantum biology pioneers whose prescient work was dismissed. In the late 1930s, Harold Saxton Burr, a professor of anatomy at the Yale University School of Medicine, sought to measure and characterize the immaterial biomagnetic field associated with living organisms. Burr steadfastly believed that life not only exhibited electromagnetic properties, but that these same properties represented an organizing principle that shaped the growth and development of cells, tissues, and organs. His research in 1938, employing the then state-of-the-art electrical measurements, provided indisputable evidence for his belief. At the same time, American inventor Royal Rife independently verified and advanced Burr's hypothesis. Rife created a beam machine that would weaken or destroy pathogens and cancer cells by focusing specific constructive or deconstructive interference energy fields on the cytoplasmic chemistry. The experiments of both Burr and Rife revealed that the appearance of certain diseases such as cancer is preceded by a measurable change in an organism's energy field. Most importantly, by simply altering the frequencies of the cell's energy field, they could ameliorate these pathological disturbances. By frequencies. I'm just going to open that iced tea. Apparently they got rid of Nestea or something. Nestea is gone. So I'm trying the new fused iced tea. And by no means is this any sort of advertisement. I'm just trying it, that's all. The scientific focus on describing the chemical nature of living organisms led conventional materialist scientists to bury thought-provoking findings of Burr, Rife, and many others that show that life is intimately connected to invisible energy fields. Recent studies are harder to ignore because though they demolish long-held assumptions, they also use convincing state-of-the-art technology, including advanced genetic engineering techniques, new microscopes with super high resolution and sensitivity that provides images of living cells, and fluorescent markers that scientists can attach to proteins to clearly delineate their movements. These instruments are not only providing insight into how quantum mechanics affects everyday biology, but also are motivating researchers to better understand how biological systems use quantum mechanisms, develop new, efficient technologies for organic synthesis and energy capture. One bioengineering innovation known as optogenetics, a technology by which cells can be programmed to respond to specific environmental frequencies, make it clear that cellular molecules especially proteins, represent physical nanodevices whose behavior can be controlled by impacting their inherent electrical and magnetic properties. Properties using applied environmental energy fields. In optogenetics, engineered DNA blueprints that combine a light-sensitive pigment molecule with a specific function controlling cell membrane receptor are introduced into nerve cells via a virus. Excuse me. The infected DNA then encodes the engineered receptor protein that is naturally incorporated into the cell's membrane. When exposed to a specific frequency of light, the new receptors on these infected cells are activated, which in turn engages the cell's function. Despite this groundbreaking work in quantum biophysics, and thanks in part to the vast resources of the pharmaceutical industry, an entity that thrives on sales of chemical signals and abhors drug-free energy medicine, the conventional world of medicine is still essentially ignoring the role of energy in shaping biological expression. When I first wrote this chapter 10 years ago, I said that though the medical establishment would eventually join the quantum revolution, it would have to be dragged half kicking, half screaming. Sadly, they're still kicking and screaming. In his lecture in 2000 at the 2004 Lindau Nobel Laureate meeting, Nobel Prize-winning University of Cambridge physicist Brian Josephson said that the scientific establishment suffers from pathological disbelief, a condition he described as, "I wouldn't believe it even if it were true." At the beginning of the lecture, he included the following admonition: warning. Readers may find some of the ideas in this lecture disturbing. They may conflict with various deeply held beliefs. Josephson, that's a weird one, Josephson. Josephson recalled how the topics of meteorites and continental drift were originally and vehemently dismissed by scientists as impossible phenomena. After a long period of time and the acquisition of abundant evidence, science finally caved and accepted the impossible as real. Josephson has also questioned today's negative scientific attitude about the validity of homeopathy, a more than 200-year-old alternative medicine system that is routinely dismissed by the medical establishment as quackery. The fact that homeopathy is favored by the British royal family hasn't stopped the British Medical Association from describing it as witchcraft. When asked by New Scientist editors how he became an advocate of unconventional medicine, Josephson said it was when he saw French immunologist Jacques Beveniste present his research at a conference. Beveniste published the first research article to validate homeopathy in the prestigious journal Nature. Josephson noted that Benveniste's presentation provoked irrationally strong reactions from scientists. He adds, "I was struck by how badly he was treated." Another Nobel laureate, Luc Montagnier, French, me and French don't jive, yo. The French virologist who won the prize in 2008 for discovering the AIDS virus has studied homeopathy and lauded Benveniste as a modern Galileo. Benveniste was attacked and demonized because he investigated a subject that the orthodox medical and scientific community had dismissed as a metaphysical oddity. Because homeopathy relies on highly diluted remedies, conventional scientists have disregarded those remedies out of hand, arguing that the homeopathic solutions are so dilute that they no longer contain any of the original signal molecules. In response to this critique on dilution, Montagnier told Science high dilutions of something are not nothing. They are water structures which mimic their original molecules. Additionally, what Josephson calls the simple-minded assessment that fluid water molecules cannot have a structure to maintain information is now valid. or is now invalid. This limited thinking does not include new insights offered by the science of liquid crystals that reveal how a flowing fluid, such as water, can maintain an ordered structure over macroscopic distances. These new insights nullify the standard refutations of homeopathy put forward by naysayers. While Big Pharma and closed minds prevent some scientists from jumping on the quantum bandwagon, the public leads the way into the frontiers of energy medicine. A National Health Interview survey found that 83 million American adults spent $38 billion on complementary and alternative healthcare in the 12 months before the survey was published in 2007. Wow, that's a lot of money. While there are no comparable, more recent statistics, anecdotal evidence points to the public's continuing enthusiasm for remedies outside the traditional biomedical model. And I think one of the biggest reasons for that is one, there's way too many side effects listed on the drugs that we're given. And two, like he mentions in the book, those drugs aren't spot specific. Like he was mentioning about histamine going to the site and causing an inflammatory reaction. When you take an antihistamine, it affects the histamines throughout everything, even the ones that are in your brain. So medicine isn't medicine affects our whole system. It doesn't just affect the area that we need it to. For example, the science of how transcranial magnetic stimulation, TMS, alters cognition, reported in the first edition of this book, is now being employed by lay audience enthusiasts who are building or buying electromagnetic field stimulators to enhance brain function. The new version of this technology, referred to as transcranial direct current stimulation, TDCS, sends small amounts of stable electrical current into the scalp. Depending on the region of the brain being stimulated, TDCS can influence neural activity that results in increasing attention, expanding memory, improving visual abilities and mathematical skills, and alleviating symptoms of depression. Wow. The public's use of electrical stimuli to influence brain functions, a modern version of radiesthesia, is still relatively fringe, yet it is a growing field of interest as demonstrated by increasing participation in an online do-it-yourself zapper, brain hacking message board moderated by 22-year-old Nathan Whitmore, a neuroscience researcher at the National Institute on Aging and advocate of do-it-yourself brain zapping. The quantum biophysics revolution is well underway, and yes, it may be televised. Wow. Okay, so we'll leave it there. Excuse me. And we're on chapter 5. We're on chapter 5, everybody. So, yeah. Wow. There's so much out there. And new information. The ability for us to search out information using the internet and now even having AI. What do you want to know? Right? What do you want to know? But the thing is what we do know, how do we know that is true? Because honestly, we don't know nothing. We have no idea about. We may know a little bit, like this much. But there's so much to know that it gets a little overwhelming about how much we don't know. But you can't dwell on that. But anyways, so there you go. Another podcast on the biology of belief by Dr. Bruce Lipton. And I just want to thank everybody for tuning in. Thank you, and you. Maybe there's only one of you in the room, but anyways. Thanks for tuning in, and we'll catch you at the next one.